Hello everyone and welcome. In this video, we will explore the function, types, and key aspects of regulation of gene product synthesis to have a deep understanding of this topic. But before anything else, let me introduce myself first. My name is Relan M. Alivio, a BS Biology second year student. Now, let's begin with a question. What is regulation of gene product synthesis? Ah, Regulation of gene product synthesis is also known as gene regulation. It's the process by which cells control the amount and timing of gene expression. This complex process allows cells to adapt to changing environments, respond to signals, and handle damage. There are four key aspects of gene regulation, but Ms. Oliveros will explain this later. Types of gene regulation. There are two types of gene regulation, the negative regulation and positive regulation. When we say negative regulation, it means inhibiting transcription through repressors that bind to DNA or blocking RNA polymerase. However, positive regulation, it means enhancing transcription through activators that bind to DNA, promoting RNA polymerase activity. Importance of gene regulation. First is cell differentiation. It means the gene regulation enables cells to specialize and perform specific functions. Second is response to environment. It means that cells can adapt to changing environments by regulating gene expressions. And lastly, disease prevention. It also means that malfunction in gene regulations can lead to diseases such as cancer. The regulation of gene product synthesis or gene regulation serves several crucial functions. First is cellular adaptation. In cellular adaptation is responding to environmental changes. It means that cells can adapt to changing conditions by regulating gene expression. Second is developmental process, which means the gene regulation enables cells to specialize and perform specific functions. Second is response to environment. It means that cells can adapt to changing environments by regulating gene expressions. And lastly, disease prevention. It also means that malfunction in gene regulations can lead to diseases such as cancer. The regulation of gene product synthesis or gene regulation serves several crucial functions. First is cellular adaptation. In cellular adaptation is responding to environmental changes. It means that cells can adapt to changing conditions by regulating gene expression. Second is developmental process, which means that gene regulation plays a key role in cell differentiation and development. Maintaining cellular homeostasis. First, in controlling gene expression, it means that regulating gene expressions help maintain cellular homeostasis and prevent damage. Second is respond to stress. It means that cells can respond to stress by regulating gene expression. Preventing disease. First, preventing uncontrolled cell growth. It means that this regulation of gene expression can lead to disease like cancer. Second is maintaining genome stability. What does it mean? It means that the gene regulation helps maintain genome stability and prevent mutations. Other functions. First, cell specialization. It means that gene regulations enable cells to specialize and perform specific functions. Second is responding to signals, which means that cells can respond to signals from other cells or the environment by regulating gene expression. Lastly, and third, is evolutionary adaptation. It means that the gene regulation can influence evolutionary adaptations by allowing cells to respond to changing environments. In summary, the regulation of gene product synthesis is essential for maintaining cellular homeostasis adaptations to changing environments and preventing diseases. Studying gene regulation is crucial in various aspects of our life because it helps us understand and how genes are controlled and how this control affects cellular processes, development, and diseases.
Good day everyone, I am Stephanie Oliveros and we are going to tackle what is regulation of gene product synthesis. Earlier, my partner Roland Alivio discussed what is gene regulation or known as gene expression. So now, let's proceed to gene regulation can occur at various levels. So first, we have transcription, translation, post-transcription, and lastly, post-translation regulation. In this video, we are going to talk about transcription and translation. And here is just a basic overview of these two processes. Transcription is the process by which DNA gets converted into mRNA, also known as messenger RNA, and translation is the process of converting the information stored in messenger RNA and using it to build a protein. Now, if you recall, which organ now is used to make proteins? What would you say? The answer is the ribosome. The ribosome is the site at which proteins are manufactured in the cell. So we're going to talk about that shortly. But let's go over transcription first before we talk about translation. Transcription occurs in three steps: initiation, elongation, and termination. Now, during initiation, RNA polymers binds to the promoter region of DNA. Now, what is the promoter region? The promoter region is basically a short sequence of DNA, and eukaryotic DNA. It's TATA, AA, also known as the data box. Now, this sequence is located 25 nucleotide upstream of the sites where transcription begins. The next thing that RNA polymers does is it causes the two DNA strands to separate, and during elongation, it begins to add nucleotides to the growing mRNA started that we see here. Thus, RNA polymers synthesize mRNA starting from 5' end going to 3' end. However, RNA polymers it reads the DNA strand in the 3 to the 5' direction. Now, there's two strands that you need to be familiar with. The first one, the one that used to synthesize mRNA, it's called the template strand or the antisense strand. So, that one in which RNA polymers is acted upon the other one which is not used. It is called the non-template strand, also known as the sense strand. Some textbook will call it the coding strand because its sequence matches up with RNA except the fact that uracil is found in RNA but thymine is found in DNA. Now, during the last steps of transcription, which is the termination step. The RNA polymers molecule, the mRNA strand, they all separate from the DNA template strand. Now, in this step, the poly A polymers enzyme, it caps the three ends of the mRNA strand, and this is known as the poly A tail. Now, also during the beginning of transcription, the five ends is also capped, and the reason of this is to protect the mRNA strand from being degraded by a certain enzymes. At the end of the termination step, during transcription, DNA has been used to create a pre-messenger RNA strand. Now, this particular strand has something known as introns and exons. Introns are basically longer sequences of nucleotides that do not code for anything, so they must be removed in a process known as RNA splicing. Now, the exons, those are shorter sequences of nucleotides, and they are going to be used to synthesize proteins. And so the exons remain. But the introns, they must be removed. And so now we have a completed messenger RNA strand. Now let's proceed to post-transcription regulation. This involves controlling the processing, stability, and the translation of mRNA. In RNA processing, this includes the addition of caps and tails, the removal of introns, and the splicing of exon. The mRNA stability. mRNA can be regulated by its length, the presence of specific sequences, and interaction with other molecules. Post-translation regulation. This involves controlling the activity, stability, and degradation of proteins. The protein modification, phosphorylation, acetylation, and other modification can alter protein activity. Now, in protein degradation, cells can selectively degrade proteins to regulate their levels.
听到都变，小情歌都美得我们甜，咖啡是加菲的甜甜圈，咖啡和快乐换了你选。Let's have it again，被青涩多印颗星照片，药品和我们在最亮眼，幽暗的银河为你装点。我偏偏为你，我就想要写一首歌，赶走寒风雪，像热烈的浪潮，用笔雕刻，誓言难冻结，心房里住着你我的附加世界，永远不空缺。当爱你，我表达的更直接。你曾经听我说，认真带一点幽默，计划不小心走火，失落一瞬间浓墨，收起了漫步中你可爱的酒窝，我的收获有点微甜，爱它随便别丢落。让我说对不起，姑娘只是不理，身边都是目击者，怕你爱终身无期。哎哦哦，也让我生活不再邋里邋遢，不想让你生气，只想当人类专家。在我生气时总是可怜巴巴，但是拜托你离我左心别只会疯。You know， 脚下的时候你总是誓不罢休，不洗手汗也会归扭头。那就不迷个人最爱的那首。每当你愁眉苦脸，喜剧之王会出现，我田园永远无上限，靠在我身边。你靠近听我说，认真带